Again, it's a great pleasure to be here, and I'm really quite honored to really be the warm-up act and the first speaker in this session, right before a really impressive bunch of researchers that will come after that. So I hope I'm not gonna be too long. Okay, so what I'll be telling you today about is our new essentially platform that is built around Google Colab to be able to enable deep learning for microscopy in a very approachable way for anyone from the bioscience field. And we'll be covering a little bit what it's good at. We'll be covering a little bit what it's not good at as well. And hopefully I'll give you a bit of an overview of how this works. I'll take also a couple of minutes to give you a short demo about how this works also towards the end of this. So as Luciano said, the recent years have really seen an explosion in the number of papers related to the use of deep learning applied to microscopy and bioimage analysis in general. And a lot of those work come from the computer science field, but not just from the computer science field, also from people who are well invested into image analysis from the bioscience field. And that's really exciting time. But right before giving you a bit more details about what our platform does, I just wanted to give you a very quick overview. I mean, I took the opportunity of being the first speaker of today's session to give you maybe a very, let's say, shallow and non-exhaustive, of course, view of what I think the deep learning revolution has been about. And in fact, neural network and the concepts of neural network is not a new concept at all. It's existed. It's been kind of thought through already 70 years ago, right, from back in the 50s, and had remained a bit of a mathematical oddities at the time, up until maybe the 80s and the early noughties as well, where backpropagation and the use, basically the widespread use and availability of GPUs have made the use of neural network a lot more practical. And but it's only from, say, the 2012 and 2015 with AlexNet and the building block, which is UNet, the invention of those neural networks have really kind of showcased the potential of deep learning and neural network in particular for image analysis, for image classification in the case of AlexNet, but for, say, image segmentation and image to image task, image task analysis in the case of UNet, and here showcased from the point of view of image segmentation of electron microscopy data. And then, AlphaGo in 2016, and then past couple of years, as I've been saying, I've seen just an enormous amount of works, neural networks, and tasks being developed around, helping essentially the biosciences understanding their images. And so, as Luciano mentioned, CARE has been really a seminal work in terms of how it spread across the field very quickly, and a number of other methods have also been developed. And that's obviously not an exhaustive approach of an exhaustive list of what's been going on. That's already outdated, even if that's just a couple of months old. But I'll just start by giving also an overview about what the fuss is about deep learning. And the first thing to take a look at is how classical algorithms work. And if we take, for instance, the example of, say, image denoising, which can be performed as well via classical algorithm, the typical approach would be to handcraft a number of mathematical rules that can be applied to a noisy image to obtain a denoised image. So that could be, say, a Gaussian filter or any other kind of filter that you might think about. Other approach, non-local means, and so on, can also be used. But the idea is the time is spent on handcrafting mathematical functions that allow us to go from the input to the output. In the case of deep learning and machine learning also in general, the idea is completely turned upside down, because we do start from the data. And in this particular case of image denoising, for instance, we start from a bunch of noisy image and typically equivalent denoised image in the specific case of supervised learning. And those bunch of images are then fed into a blank neural network architecture, which can then learn automatically how to perform that task, that image analysis task. And so once the trained model is obtained, it's as simple as feeding new and similar type of data into the neural network and then obtaining the denoised or the prediction of the denoised image equivalent in the output. And this is what we want to use it for eventually. So the training is only a prerequisite step for getting to the trained model to actually do what we want it to do. And so with this very general and very shallow overview, I'd like to highlight a couple of extra points. Is deep learning can, in principle, approximate pretty much any transform. So I've not put any restrictions in terms of what can be fed as input and output. And so that makes deep learning extremely versatile, essentially. So that means a very large number of image analysis tasks can be performed. I'm only presenting image to image kind of transforms, where many other type of approaches can be taken. And secondly, on top of its versatility, it can also massively outperform classical algorithms. And that's really quite important, because this is what really makes the field of deep learning applied to microscopy extremely exciting and extremely promising. And that's all upsides of it. The downside of this, at least from the uptake of deep learning to microscopy from the bioscience field point of view, say biology lab and maybe in the clinical context as well, it still requires today a significant amount of experts' computational skills, as well as access to high computational power, and in particular GPUs, as I've mentioned earlier. And so that's a clear downside to this approach. And just to highlight one important aspect of training in deep learning microscopy is the fact that it typically embeds structural priors. And one very elegant way that I like to highlight this is through this really nice work from W. Murner's group, published this year in Biomedical Optics Express, where they looked at how deep learning can recover a specific structure. And for instance, looking at here on the top row, the ground truth images, the bottom row here showing sparse sampling of those exact same structures. And what they did in this work is to try to recover the ground truth images from the sparse sampling, but by training deep learning algorithms similar to CARE and so on, to actually recover those structures, but only when they were trained on specific types of shapes. And so if the deep learning algorithm was essentially trained only on rectangles, then what the predictions will find is a bunch of rectangles all over the shop, right? And so that's a clear limitation, is that it means that whatever was fed at training stage will therefore be found at the prediction stage. And so similarly, if a neural network was trained only on triangles, despite the fact that the underlying structure that it's trying to recover contains all of those sort of shapes, only triangles are recovered. And so what I wanted to highlight from by showing you this work is the following, is that it is absolutely, in my view, essential to train on your own data, to retrain to some extent on your own data for deep learning algorithms to perform reliably. And so what this essentially means is that although there's a wide acceptance of pre-trained, or say wider and wider acceptance of pre-trained models, those still tend to or can lead to the risk of obtaining artifacts that are, however, unknown to the end user eventually. So even for people interested in the bioscience field in using deep learning microscopy, I suggest, and I think this example highlights it pretty clearly, that deep learning needs to be trained on your own data, or to retrain at the very least on your own data. And so that highlights the need for the use and the mere existence of platforms which allow you to actually train and use the cloud computing, for instance, to be able to retrain and restructure the neural network for your own purposes. And so many tools are obviously available to do this, but what we wanted to create in the lab is an entry point for anyone to be able to get started in deep learning microscopy. And so to basically be able to overcome this hurdle of feeling like retraining or training a neural network might feel like maintenance on the International Space Station. And I like to show this actually, because this is Michael Hopkins, an astronaut that was on the International Space Station performing some maintenance on the ISS, and that from a bioscience point of view, training and setting up the hardware and software architecture necessary for training a neural network might feel a little bit like this at first sight. So without further ado, let me introduce you to our approach to try to tackle this issue. Is to essentially use the already available Google Colab platform online that's owned by Google, of course, a private company with the downside that come with it. But that still is really quite useful from the point of view of getting started. And so here's how our architecture, our platform work. It always starts from your own computer, so your own analysis workstation where you'll have typically, or you'll have somehow the training data and the new data. The first step is to actually push that data to a Google Drive. And what our platform really essentially provides is a set of notebooks, Google Colab notebooks, that will then be able to connect to Google Colab, and that will be completely self-sufficient to be able to perform neural network training directly on the cloud, exploiting Google Colab's free resources in terms of memory and GPU and access to computational power. So training as a first step, but also validation of the neural networks and the trained models, as well as running all the predictions necessary to then recover all the results from the new data as well as the trained model. And so you see that by being able to recapitulate the whole workflow of starting from a bunch of data that was previously created by the user, performing training, validation, prediction, and then recovering the data, we've actually gone through the whole pipeline, the whole workflow of deep learning to be able to exploit it as best as possible. And so the important thing is, because all the computational burden is now performed on the clouds through the cloud processing provided by Google Colab, then no requirements are necessary onto the user's machine. And so that has a number of advantages. Is that currently with the way it's set up, our platform called ZeroCostDL4Mic provides basically a free cloud-based access to GPU for deep learning, a simple user interface, right? So there's no coding required, but all the codes are very easily modifiable and accessible to anyone who wanted to go check and make any modifications to the code we've provided. And importantly, from the intention we have behind this platform, is that it's a single platform for training, prediction, and quality control. And so that's also quite an important step that needs to be highlighted, and we'll get back to that in a minute. So from the point of view of the user, again, this platform will only take the shape of a web browser page, essentially, a Google Colab web page. And so what this means is that it will only require access to a web browser for you to be able to run the platform in the first instance. But what can the platform actually do? And I should highlight something absolutely clear at that stage, is that what we provide, or the added value that our work actually provides, is basically a consistent set of notebooks that are based on currently state-of-the-art image analysis neural networks that were developed by great computer scientists around the world, and we've re-implemented those neural networks to be able to be integrated within our platform. So we haven't created any neural network at that stage. And so what can the platform actually do? A number of image analysis tasks, and one classical image analysis is that of segmentation and object detection. And so for this, we've implemented a classical units from Rannenberger from back in the days, but also the 3D version of it, and which we've highlighted on electron microscopy data. I would also like to say that we've provided a bunch of test and training data set for anyone who wanted to try playing with this data set. We make them available for you to have a go at training and running predictions on those networks. And so what you see here is segmentation of a mitochondrial network into 3D electron microscopy data. And the green being the ground truth and the purple being the prediction on this test data set. Other kind of segmentation that we've implemented is an instance segmentation approach provided by Stardist, essentially built around the idea of segmenting round or star-shaped objects, hence the title, but typically round cells or nuclei in cell migration assay from fluorescence data microscopy, as shown here, for instance. And also, we've also implemented YOLO V2, providing object detection from here demonstrated on also a cell migration assay from bright field images, where we've identified 3 different classes: elongated, rounded, and dividing cells in this movie. Another kind of image analysis task is that of image denoising, and as we've discussed earlier, we've re-implemented the CARE network, allowing us to denoise and also remove a number of artifacts from raw images right here. Here we're looking at structured illumination microscopy data, highlight, and you can immediately see from the raw data on the left that you have the typical, say, hexagonal type of noise pattern that you see in structural illumination microscopy that we've managed to actually remove from CARE denoising type of approach. We've also implemented noise to void as a self-supervised denoising approach, also demonstrated it on, here in this case, 3D mitochondria dynamics of live cells right here, of live cell data set. So showing, of course, on the left the raw data and on the right the denoise data. We've also implemented a couple of what we call image-to-image translation type of approach, and I'll show you a couple of examples, such as that of the inference of pseudofluorescence images from bright field images from the label-free prediction or FNet that was published a couple of years ago by a group from the Allen Institute. An extremely powerful approach, in my opinion, where essentially chemically specific type of marking or labeling can be obtained from a label-free image, and so that can alleviate to some extent the need to even fluorescently label certain samples. So that's really quite a game changer in fluorescence microscopy and general microscopy in this particular case. And we've also implemented pix-to-pix as a GAN approach to microscopy type of data set, and we've demonstrated how it can be used here in this particular case, doing the prediction of how a DAPI marker would look like typically from an actin label. And so here, what this means is because the actin label sort of has sufficient information within itself, and you can already start guessing where the nuclei are from this, from the raw data. then it's actually relatively straightforward for Pix2Pix to actually create fake nuclear signal and recover a pseudo DAPI type of signal from here. Again, here, alleviating the need altogether to have nuclear markers in certain specific fluorescence microscopy studies. I just wanted to also show you this really fun study that we've also created, and let me just pause this for a second, where we've combined a number of those networks back to each other, as well as hooking it up with more conventional type of image analysis. And let me show you what we've done right here. The raw data, and the only data that was actually physically acquired, is a LifeAct RFP, so an actin network, a live cell actin network fluorescence time series, as shown right here. And so we used Pix2Pix to now actually infer a fake cDNA or DNA marker from that LifeAct RFP dataset, therefore obtaining a pseudo two-color image and a pseudo two-color time course dataset. And this essentially the nuclear marker, the fake nuclear marker obtained via Pix2Pix, can then be fed into Stardist for instant segmentation, and therefore the analysis and the identification of each individual nuclei in the image. And this output can itself be fed into TrackMate, a common and very popular type of approach for single particle tracking, currently also implemented in Fiji. And so if I run this, actually, this whole pipeline allowed us to really start from a simple actin dataset and obtain a fully quantitative single nuclei tracking from this cell migration assay, with all the information that comes with it. And there's a little protocol that's, by the way, available in this one F1000 research paper down there as well, if you're interested. But I promised you I was just going to get very quickly to the idea of model validation. And so the bottom line question here is, can you trust your model? And in fact, if you have a source image, say a noisy image of here, mitochondrial organization, here TOM20 confocal dataset, and you try to run, say, care for denoising this particular image, you obtain a prediction output. Can you trust this output? Can you actually believe that this does not contain too many artifacts, or this does not produce erroneous type of results? The most straightforward and intuitive way to actually validate this is to actually compare it to a set of ground truth data and to so-called target data, of course. And so what we've implemented in the platform that I've shown you just right here is a couple of ways to do quality control, one of which is simply by looking at essentially the unsigned difference between those two images, showing us the error, and a normalized error gives us a quantitative idea about how well it performs, and that also allows us to do a network optimization. And we've also, because here in this case, the model, the errors are actually reasonably low, then that confirms to us that this particular prediction actually worked really well. And the target image can also be compared to the prediction by also looking at structural similarity type of metric. Also a very common type of metric that can also be displayed as a map. And a high, in this particular case, a high structural similarity will highlight a very high quality a model prediction in this case. And so in this particular case, we get a good validation of this trained model. And so just to wrap this up, of course, there's a catch. Every single approach will have a catch, and in particular, there's no such thing as a free lunch. And similarly, there's no such thing as free access to unlimited cloud computing, right? beyond the fact that this Google Colab is run by Google, it also has a number of physical limitations that I wanted to highlight to you today. So it's absolutely free, so it costs nothing to use, but there's only a small available, which are reasonably good, but already outdated in the current market, and a number of maybe more stringent restrictions here are the fact that the session times are only 12 hours. RAM available is only 12 gigabytes, and the disk space available in the virtual machines created in the cloud are only of the order of 30 gigabytes. And so the bottom line about this is that essentially there are a number of limitations of Colab, which means that we can only run training for a limited number of epochs, right? Due to both the RAM availability and the session time. But also the RAM availability will will restrict the amount of training data that can be used in those particular cases. And so we've run a little bit of rough analysis or rough estimation of what this means in practical terms to have those limitations in terms of RAM and disk space. And in particular, I will highlight a couple of tables where you see on the left all the different networks that we've implemented in the platform and the type of data that we've tested it on. But the bottom line that is important to take away from this table is that only maybe a few hundreds to a few thousands of paired images can be used due to the limitations of either RAM or Google Drive disk space in some of those cases, right? And so you see that some of those numbers, although they've not appeared to be limiting for us to create high quality models, such as all the ones that I've shown you, that have produced the results that I've shown you so far, this can be a limitations, of course. And those numbers might seem also reasonably small for more classical Google, image, say vision type of approaches. And we've also run a couple of analysis of what Google Colab limitations has to put on us on prediction stage. And so at that stage, purely the number of predictions that we can run isn't limited by the inference rate or anything like this. It's really due to the Google Drive disk space, essentially. And that limits us the number of stacks and images that we can run in any single session of Google Colab analysis. So all of this, just to give you an overview, and the final words on this is that we've really designed a platform that is good at a certain number of things, but maybe not as good for a number of other things. So what we think is good at is that it's really good at prototyping image analysis workflows. If you want to say test whether Google deep learning is good for a particular data set you've got before you start investing time and maybe money into specific platforms, that's a great place to start. It's obviously good for small to medium-sized projects, but beyond that, it's a little bit trickier, and that requires some investment or some tweaking around. And it's also good for short-term projects that don't require maybe a permanent deep learning infrastructure locally. And so that's especially true for maybe smaller research institutes that don't have imaging platforms that could set deep learning infrastructure locally. And I think that's one of the reasons why this has been popular since we've actually come up with this platform, is that it's a good resource for deep learning enthusiasts and for students to learn about deep learning and start getting their hands dirty onto what deep learning can do for a specific type of image analysis. What it's not so good at, and I'd like to be clear about this, is that it's obviously a lot more difficult to run large-scale projects, say projects that will be using over 20 gigabytes of data for training, for instance. And so that can be a limitation. Also for long-term analysis pipeline, right? Similarly to the idea of having a DL infrastructure, some projects might benefit better from a pure deep learning infrastructure. And also for generating a highly general model, also purely due to the fact that, highly general models will obviously require a very large amount of data and very long computational times. And so just to give you a final look at what I've shown you today, we've created a platform based on Google Colab called ZeroCostDL4Mic, and it's very compatible with working from home. And we've also got a number of very well documented pages that are all open source and that you can take a look at if you're interested. And we've implemented a number of networks, and that's an ever-growing endeavor. And we've also got a little preprint that you can take a look at if you're interested in getting further details. And just to wrap it up, I'd like to acknowledge all the authors that have contributed to this one way or another, and that includes the deep learning community, developers and developers, beta testers, and so on, that have created the networks, tested them, and also helped us to really get something hopefully useful to the bioscience community. Luciano, do I have a couple of minutes to do a very short demo?
Yeah, if you keep it brief, I think we can. We would like to keep each session about 30 minutes, so
okay.
we can go much further. But I definitely want to see a demo, of course.
Okay,
then I
can use this slide then. It's just to use this slide essentially. Okay, let me just very quickly then show you what is going on, and that'll just give you just a very short flavor of what this is about. If we take a quick look at our GitHub page, all our notebooks, the notebooks that we provide are currently deposited into this table, and from which all of the different Colab notebooks can be open. Let me actually open something sensible. Let's take a very quick look at Stardust. And to make it really easy to use for as really a starting point, and we like to call it a gateway drug to deep learning microscopy, because that's an easy way to get started and to consider whether it's worth further investment. But if we take an example as a Stardust notebook, we actually recapitulate here in the table of contents complete workflow of deep learning, from initializing of the session, installing the different dependencies, setting the parameters for training the network, doing the quality control from evaluation and the SSIM, and doing some other quantitative and qualitative analysis of model performance, all the way down to prediction and using the actual trained model. And just to give you a rough idea, I've just opened this page, and so can you in a couple of seconds. If you want to start using it, it's as simple as running the notebooks online. The connection to Google Colab and the cloud platform is done absolutely immediately. And keeping in mind the limitations I've highlighted to you, we think that this provides a real way to get started for a number of bioscience image analysts and anyone interested in microscopy in general. And so I think I'll stop here in the interest of time then. I'll happily take questions, and thank you very much for your attentions.
Love the slide. Tremendous, really great work. This is, I think, one of the more popular platforms now in the open source community for training and applying deep learning models. And that's really definitely a hurdle to the community, to have an easy to use a platform like that. And this one not only is a Google Colab, but it's has a low cost or no cost in some cases, and it's accessible and open source. So that's really great effort. Congratulations.
No,
I
thank
do
you
have
very
a
much.
question. I do find the validation part to be really a critical portion to acceptance and broader use. What can you comment there? Because you showed some really obvious fail cases with your squares, and there's a few reviews that show some examples of text that can be completely, mashed up and created into another way that is not real. So what can you say and comment about quality control of the images, especially when it's a generative approach or a completely unsupervised approach?
But you're absolutely right. It's essential, and then I would never use a trained model that hasn't gone through some sort of quality control. And in fact, what I say to everyone who get in touch with me regarding exactly this issue is find a way to generate some ground truth data, right? There is no ways around it. Without ground truth data, there isn't real obvious ways to really build a good trust in the outputs, and especially from a user's perspective, to be able to visually and quantitatively have an idea about what's going on, and especially judge what kind of artifacts appear, and maybe whether those artifacts are relevant or not to what is being studied at stake. It's also a way to get an understanding of whether the artifacts that are generated in some ways can be dealt with, or can be somewhat are acceptable or not for the particular maybe downstream analysis. But it's important to have a really good idea about the performance there, that's for sure.
Yeah, thank you. I wonder if there are other questions from the people attending today. While there are no questions, I will just thank you for correcting the group leader topic. I'm pretty sure that very soon you will be, but right now I double checked. I mean, and so Romain is currently a recipient of the MRC Skills Development Fellowship, and also a Lido Associate working there at UCL. If you have any group leader positions open, you should definitely contact him, recommend it. And
thank you for the kind words.
So if there are no other questions for Romain right now, let's progress to the next. Oh, there is one question just came up. You can probably see it in the chat. I'll read it out loud for those that can't see the question. Okay. Thank you for the presentation. Does your platform use Google Colab TPUs? Are they faster?
So we've actually spent quite a bit of time on trying to get TPUs to run. They do run, and in fact, it seems that Google is really trying to use their Colab platform as a way to promote their own TPUs, of course, and that's absolutely fair. But in our hands, it has always been consistently slower than the GPU that they provide. There might be a few additional tricks that we've not investigated yet to get them to perform better, but currently that hasn't been as successful as the use of GPUs. So for the interest of getting a platform that works, we've stuck to GPUs to a large extent. Yeah. But it's a fair point, and we're also wondering whether there's something we're missing in terms of how we exploit the TPUs. And I'd happily hear from anyone's thought on this, by the way. Yeah.
Yeah, it may require changes to both the networks that are set up and the hyperparameters and all of that, so that they are optimized for TPU usage. So it may not work completely directly, but yeah, I do agree. Google does try to get everybody onto the TPUs.
Yeah. The gateway drug hypothesis.
I see one more question from Uri Manor. Yep. Is it possible to load a pre-trained model? If so, couldn't you just train a model on more data by dividing into separate sessions?
So in fact, Uri, this is exactly what we do. It's an obvious step to allow for pre-trained model and then run across sessions. But in practice, only very few of the networks that we've implemented require this, apart from the very large, say, 3D UNet segmentations, or for FNet, which also requires really long training times of the order of tens of thousands of iterations. But yes, of course, you could do this. With the downside that restarting a full session with the way we currently save the models, which does not completely save all of the state of all the optimizers, there is a form of training information that is lost along the way. So it isn't strictly equivalent than as running longer sessions. But we've shown that using transfer models as we've implemented it also helps with this. Yes, absolutely.